So today's topic is integration of metabolic pathways and regulation. So in that the subtopic is coordinated regulation of glycolysis and gluconeogenesis. So when we talk about the metabolic pathway, the flux is very, very important. What is meant by flux? It is the flow of metabolic pathway. If you look at this image, here the glucose is converted into glucose 6-phosphate. And from there, pyruvate is synthesized and the other molecules are produced. Lactate, alanine, and the TCA intermediate, acetyl-CoA. So this is one flux. Okay. So if you look at the same glycolysis, you have another pathway leading to the gluconeogenesis, which means from the pyruvate, glucose 6-phosphate is synthesized. And from that, again, you can get the glucose. So this we can call it as a reverse flux. Okay. So the glycolysis may be a forward flux and gluconeogenesis may be a reverse flux. Similarly, if you look at during the glycolysis process itself, if we have excess glucose, it is converted into glycogen. That process also we have seen, that is glycogenesis. And the reverse, this glycogenesis is going parallelly, so it can be called as a parallel flux. There again we have a reverse flux. Glycogen is broken down to glucose 6-phosphate. So when there is a requirement of the glucose and the glycolysis, there is no glucose in the diet and the energy is required. In that case, the stored glycogen can be broken down by glycogenolysis, that also we are familiar. And this we can again call it as a reverse flux. And it is going to the TCA cycle. From there, energy is produced. One more parallel flux, you can see the pentose phosphate pathway. It is also starting from the glucose 6-phosphate of the glycolysis. It produces pentose phosphate and it is a parallel flux. So in the carbohydrate metabolism itself, if you look at different pathways are there: forward, parallel, and reverse flux. And these flux can be determined by the main step in any of these metabolic pathway, the rate determining step. In glycolysis, the first step, which is catalyzed by hexokinase or glucokinase, is called as rate determining step. Similarly, glycogen, the first step is called as the rate limiting step or rate determining step. In pentose phosphate pathway also the same, glucose 6-phosphate conversion is the rate limiting step. For each flux, the first step mostly is the slowest step in the network of the reactions, and it will be near the beginning. Mostly it is near the beginning, and it can be regulated by feedback inhibition. When the product level is high, this pathway can be inhibited or this flux can be inhibited. So that step, the first step, is called as rate determining step. And the metabolic pathway, the feedback inhibition or for feedback suppression, all that can be carried out by mainly by two mechanisms. One is by covalent mechanism. Covalent mechanism is addition or removal of a chemical bond, or it can be carried out by non-covalent method. So all these allosteric regulations, hydrogen bonding, electrostatic interactions, or via van der Waals force. So this regulation, allosteric regulation, is called as the non-covalent allosteric regulation. And this metabolic flux can be changed. The turnover of this metabolic flux is regulated based on the stoichiometric reaction model and the utilization rate of the metabolites, as well as the transport of these molecules across the lipid bilayer. So though you have the metabolite or the precursor like glucose, it should be transported through the glucose transporter into the cell. So it is, the flux is also depending on the availability as well as the transport of this molecules or precursors. Utilization of the metabolism, that is also an indicator showing the scarcity or demand Okay, so based on this, the metabolic flux changes, it can be regulated. Okay, availability of the precursor and the demand, as well as the translocation process, whether it is translocated or transported efficiently, all that will affect the flux of the metabolic pathway. It can turn over the metabolic pathway. And if you look at the diagram image given below, so there you can see not only the carbohydrate metabolism, the other metabolism also is integrated, and it is also giving a flux. So carbohydrate metabolism, the main pathway is glycolysis, and into this lipid will join, as well as the protein through amino acid metabolism also will join. So the flux is also integrated with the metabolism of all the biomolecules. Okay, not only the carbohydrate metabolism we have seen earlier, now the protein and lipid metabolism also is integrated into the flux of carbohydrate. So integration of metabolic pathway of different biomolecules can be explained in this. This flux, whether this production, the pathways towards the glycolysis, or the pathways directed to the lactate production at anaerobic condition, or it is going for pentose phosphate pathway, so that can be traced. Okay, that can be traced. This flux can be traced. How do you trace it? We have to label the molecule which we are analyzing. For example, if most of the molecules are having carbon in it, so particular molecule, you just do the radio labeling with the carbon C13. Okay, so that flux from the substrate where it is going, whether it is taking the glycolytic pathway or whether it is taking the pentose phosphate pathway, that can be traced by analyzing the radioactive emission and the mass of the substrate. So this metabolic flux analysis and the control point will be the one which is directing to different types of metabolic pathway. So this control also can be traced using radio labeled experiments. So many such experiments are going on. This topic, MFA and MCA, will be detailed in metabolic engineering part, so that the flux can be altered. That is called metabolic engineering. Metabolic flux analysis, metabolic control analysis will give you an idea to alter the metabolic pathway in order to get high quantity of particular product rather than the other metabolic pathway. The other metabolic pathway can be just simply, it can be suppressed, so that only one metabolic pathway, one flux can be activated, so that the product will be in high yield. So now coming to the regulation of carbohydrate metabolism in animals to achieve homeostasis. So homeostasis is the dynamic steady state of any cell or any organism, organ as well. And this homeostasis of all the biomolecules is essential in our body. So the regulation of carbohydrate metabolism, let us see in this slide. Main carbohydrate metabolism or anabolism and catabolism. In carbohydrate, anabolism is glycogenesis, glycogen synthesis. And the other one is gluconeogenesis, glucosynthesis. Both are anabolism and carbohydrate metabolism. Similarly, catabolism, if you look at, in carbohydrate metabolism, glycolysis, you are familiar with that. Then TCA cycle. Then glycogenolysis, the breakdown of glycogen, that is a catabolism. So these are the main carbohydrate metabolism coming under catabolism and anabolism. And these two, catabolism and anabolism, are kept in balance in order to maintain the homeostasis of the cell, homeostasis of the organ and organism as such. So this is determined and this is regulated basically by the metabolites and the availability of oxygen and so on. Demand for ATP production is the main criteria which regulate this anabolism as well as catabolism, where the flux should go, where synthesis should be promoted or the catabolism should be promoted. That is determined by the demand of ATP production in muscle. So it may increase 100-fold in response to exercise in few seconds itself. Okay, and so that will give the demand. So that will automatically enhance the glycolysis floods. Similarly, wound healing, that is another process. So that require huge amount of energy and biosynthetic precursors for the healing of the wound. So at that time, accordingly, the particular metabolic pathway will be switched on, or the floods will be enhanced. And bioavailability of the oxygen. So oxygen may decrease due to hypoxia condition. Different disorders or diseases will lead to hypoxia condition, especially the cancer, and it will lead to the decrease in the availability of oxygen. And so the delivery to tissue also will be diminished. There is another condition called ischemia. There the blood flow to the tissue will be diminished. Okay, so these are the conditions which will give the stress, and this also will alter the metabolic floods. And then the availability of carbohydrate, the source for the metabolism, carbohydrate. And if carbohydrate is not there, fat can be used. If that is also not there, protein can be used. So all these availability in the diet, okay, so that also influence the metabolic floods. So they vary from meal to meal, and all these require metabolic adjustments. So based on the meal to meal interval, exercise, or wound healing, or any other stressful condition, hypoxia, the metabolic adjustment should happen. So this will happen between meals and during starvation, and it depends on the demand and availability in the body as well as diet. So the net effect is to achieve homeostasis. It will return the organism to the normal dynamic steady state. So if you look at the image, at fed state, insulin will be predominating. Okay, so the major site of insulin action is on fuel metabolism. Here the plus indicate the stimulated, and minus indicate the inhibition by insulin. If you look at fed condition, so the plus glucose to glycogen, that is glycogen storage, will be high. Okay, so that is stimulated by the insulin, indicated by plus. Similarly, the reverse glycogenolysis, breakdown to glucose, is decreased because we are in the fed state. There is no requirement for the glycogen to break down. So that is indicated as negative in the liver. Similarly, if you look at in the skeletal muscle also, simultaneously such adjustments are happening. The glucose, the excess glucose coming to the blood system and going to the skeletal muscle, and there it can be stored as glycogen. Not only in the liver, it can be stored in the skeletal muscle as well. And this glucose goes to the adipose, and there this is enhancing the conversion of fatty acid to triacylglycerol. So the triacylglycerol is the storage form of the lipid happening in the adipose. So that is promoted. This lipogenesis is promoted by the glucose level. And then in the fasted or starved state, the reverse, almost all the mechanism is reversed. So you have the low insulin here. What are the other hormones dominating? Glucagon as well as in the muscle, epinephrine, another hormone. They will be predominating, especially in the liver. Glucagon will be predominating. Glucagon is just opposite to insulin. So the major site of glucagon action on fuel metabolism, if you look at, so it will promote the glycogenolysis. You can see the plus symbol shifted to the right side. So glycogenolysis is promoted by glucagon, whereas the glycogen synthesis is inhibited by the glucagon in the liver. And similarly, in the skeletal muscle also, this will be converted into the ketone bodies. Glucose is converted into the ketone bodies, and they will go to the skeletal muscle. And then the fatty acid metabolism also is reversed in the adipose tissue. There the lipid is utilized here. Instead of glycogen, lipid is utilized here in the adipose tissue. Now let us see the glycolysis and gluconeogenesis. The main process, gluconeogenesis, is reversal of the glycolysis, but not exactly the reciprocal reaction. We have differences. Gluconeogenesis occur mainly in the liver. And when it occurs, when there is a scarcity for the glucose, you don't have the dietary glucose, you are in long fasting. At that condition, the glycogen stored glycogen also will be exhausted. Then what is the other source? From the non-carbohydrate metabolites or precursors, gluconeogenesis will be synthesized. It is happening primarily in the liver. Gluconeogenesis employs most of the enzymes that act in the glycolysis, but not all. Not simply the reversal, it is a separate pathway, but it is sharing the enzymes and metabolic pathway of glycolysis as it is shown in the diagram. So they are not identical pathway running in opposite. That you should understand. Out of 10 glycolytic reaction, 7 are shared as freely reversible pathways. So the gluconeogenesis pathway is indicated with blue color here, arrows, while the 3 irreversible reactions in the glycolysis, they will be carried out by a different enzyme. The 7 reversible reactions are carried out by same enzyme, and the 3 irreversible reactions are carried out by a different enzyme for the gluconeogenesis, that is highlighted in blue color. In the first step, glycolysis is carried out by hexokinase, whereas the gluconeogenesis is carried out by glucose-6-phosphatase. So you know that kinase will add the phosphate, whereas phosphatase will remove the phosphate. So it is just a reversal of it, but carried out by another enzyme. Similarly, the other next reaction is same, and next reaction, it is different. Conversion of fructose 6-phosphate to 1,6-bisphosphate is carried out by phosphofructokinase in glycolysis, whereas in the gluconeogenesis, the reverse is happening by fructose 1,6-bisphosphatase. Here again, the removal of phosphate. And next conversion to the glycerol 3-phosphate and dihydroxyacetone phosphate is a reversible process, and interchange of this dihydroxyacetone phosphate and glycerol 3-phosphate is 4, 5. And from glycerol 3-phosphate, by a reversible reaction, 1,3-bisphosphoglycerate is formed in glycolysis. The same thing is shared by the gluconeogenesis. And next step, conversion to 3-phosphoglycerate is also shared for gluconeogenesis. And next, formation of 2-phosphoglycerate, that is also shared by the gluconeogenesis. And phosphoenol pyruvate formation is also shared. Now, the last irreversible reaction, phosphoenol pyruvate to pyruvate, that is carried out by pyruvate kinase in glycolysis, whereas in gluconeogenesis, it is carried out by 2 enzymes. The first one is converting the pyruvate to oxaloacetate. That is done by pyruvate carboxylase. And oxaloacetate is converted back to the phosphoenol pyruvate PEP by PEP carboxylic kinase. So in the glycolysis, there the pyruvate kinase is adding the phosphate, whereas here also it is carboxylic kinase adding the phosphate. So it is not removal of phosphate. So like this, the gluconeogenesis is not identical pathway running in opposite, but it is sharing some of the reactions of glycolysis, and it is reciprocal to the glycolysis. Gluconeogenesis is reciprocal to glycolysis. So these enzymes, mainly these 3 enzymes, are undergoing regulation. The allosteric regulation happens for these 3 steps. So the coordinated regulation of glycolysis and gluconeogenesis, it is reciprocally happening. So it is regulated for certain reasons. One main reason is both proceed simultaneously at high rate. It consumes ATP and produce heat. So in that way, if both happen simultaneously, then the metabolic pathway is uneconomic. Both consume energy. So it is called as futile cycle. So in order to avoid such futile cycle, when glycolysis is happening, the gluconeogenesis will not happen. There is no need. When glucose is there, glycolysis is happening. When glucose is not there only, the gluconeogenesis should happen. So it is not happening simultaneously. Reciprocally, it is regulated to prevent the wasteful operation of both the pathways at the same time. So that when the flux of glucose through the glycolysis goes up, the flux of pyruvate towards glucose goes down. So here you can see the turnover of the floods, and this is coordinately regulated to ensure that both don't run simultaneously to prevent the energy waste. How it is done? By targeting unique, irreversible steps in each pathway. So it can be done through the hormonal signal, it can be done by allosteric effectors, and it can be done by transcriptional control at molecular level. So in molecular biology, that will be explained in detail. So the hormones mainly glucose, glucagon and insulin. Allosteric effectors, ATP, AMP. Even the fructose 2,6 bisphosphate, that substrate is also acting as an allosteric effector. And then transcription control is molecular control. Reciprocal changes during fasting, the rate of glycolysis in the liver decreases, while rate of gluconeogenesis increase, leading to the higher hepatic glucose production. So the reason for this coordinated regulation is mainly to avoid the futile cycle as well as to maintain the metabolic homeostasis. So it is done at cellular level. Allosteric regulation is done in the glycolysis. The phosphofructokinase 1, PFK1, is activated by AMP and fructose 2,6 bisphosphate. So which is the signal which signal a need for energy and ATP production. And it is inhibited by high levels of ATP and citrate. So AMP and ATP are just opposite. So it is activated by AMP, whereas it is inhibited by ATP and citrate as well. Citrate, you know, that TCA cycle product, intermediate. It is indicating that sufficient energy is there, ATP, sufficient energy is there, so it will inhibit the glycolysis. At the same time, gluconeogenesis is inhibited by AMP. It is just opposite. Gluconeogenesis, mainly the enzyme fructose bisphosphatase is inhibited by AMP. And then fructose 2,6 bisphosphatase is prevented by glucose production when energy is needed. And so the key enzymes regulated are pyruvate kinase and the pyruvate carboxylase involved in the gluconeogenesis. Pyruvate kinase, it is the last enzyme in the glycolysis. That is the irreversible step that is regulated by allosteric effectors. Similarly, the first enzyme, pyruvate carboxylase, in the gluconeogenesis, because from the reverse, if you look at, that is the pyruvate carboxylase, and that enzyme in the gluconeogenesis is regulated by the acetyl-CoA level, which signal that cellular energy demands are met. So such regulation you can see and illustrate with this diagram. High AMP indicate that the low energy charge, and it signal the need for ATP and promote the glycolysis. High ATP and citrate, it indicate the high energy charge and abundant intermediates, so that can inhibit the glycolysis. So the enzyme regulation by allosteric mechanism is explained here. And lactate and alanine also will influence this metabolic flux. If you look at in the muscle, alanine is produced from pyruvate by transamination, and then alanine inhibits the pyruvate formation via glycolysis. So in the liver, alanine is converted back to the pyruvate. It promotes the gluconeogenesis. In an active muscle, lactate builds up. It passes through the blood and it is converted to pyruvate in the liver. Hence, gluconeogenesis is promoted, rather glycolysis. So thus part of the metabolic burden of active muscle is shifted to the liver. So the organs are having crucial role in the allosteric regulation of glycolysis as well as gluconeogenesis in a reciprocal manner. Hormonal regulation, insulin and glucagon, I showed you in the previous slide, promotes glycolysis by activating the pathway that lead to the increased fructose 2,6 bisphosphate phosphatase. While the glucagon stimulate the gluconeogenesis, its aim is to raise the blood glucose level, particularly during the fasting, by favoring the glucose production. And transcriptional regulation at molecular level, it happens, these enzymes, the rate limiting enzymes for the gluconeogenesis is produced when there is a requirement. So at the level of gene expression in response to the hormonal and metabolic signals, these enzymes are regulated, or they are generated from the mRNA. So the regulation happens at the mRNA level or at the transcriptional level.